Hello, I'm Yvonne Stapp for Science for the Public, an organization committed to bringing recent science discoveries to the general public. Please visit our website to learn more about our science lectures at Boston Public Library and check out the resource lists on a wide variety of science topics. Today, welcome to Public Science, a series on current developments in science that are shaping or reshaping our understanding of nature. This series presents scientists who are on the cutting edge of research and discovery. Today's presentation is by Dr. Chris Organ. Dr. Organ is a postdoctoral fellow in the Department of Organismic and Evolutionary Biology at Harvard University and an adjunct professor at Brown University. He's an expert in an area called paleogenomics, the reconstruction of ancient extinct genomes. He's been active in developing techniques that make that reconstruction possible. In this program, Dr. Organ explains the unique kind of information the genome can reveal.
I'm going to talk today a little bit about paleogenomics and about how genomes evolve over long periods of time. And I'd like to start today by talking about the mammoth genome. And mammoths are hairy close relatives to elephants that are completely extinct now. And yet the entire genome of a mammoth was sequenced earlier this year in 2009. And the researchers did this by using lumps of hair, surprisingly. So when they first started the project, they crushed up bone and they tried to obtain ancient DNA from bone. And they couldn't obtain very much DNA. When they turned to hair and dissolved hair, they obtained vast quantities of DNA, and this was a big surprise to the researchers. And what that allowed them to do is to obtain enough DNA where they could sequence the entire genome actually from several different individuals. And this constitutes the kind of the gold standard for paleogenomics. Here's a picture of a baby mummy mammoth, and these are often exquisitely preserved. Now, there are many, many species that are extinct, and very few as beautiful as this. And so, unfortunately, these kind of sequencing technologies, for as great as they are and as interesting as they are, can only sequence DNA from organisms about 500,000 years old. And clearly, a lot of extinct organisms that we want to know a lot about are much older than that, like dinosaurs, for example, which thrived tens, hundreds of millions of years ago. So I'm going to talk today about other ways that we can figure out parts of the genome and genomic biology and things that are long extinct, like dinosaurs. So not only are we interested in the sequence of a genome, we're also interested in things like how big the genome is. And this is analogous to wanting to know about body size for an organism. In addition to that, we're also interested in how a genome is structured. So a genome is a string of DNA molecules, and they're wrapped up into bundles and organized into things called chromosomes. And here you can see chromosomes from an emu, which is a flightless bird related to the ostrich. And you can see it has all of these little dots are chromosomes called microchromosomes. And these are an aspect, a characteristic of the bird genome that we don't have. Mammals don't have microchromosomes, yet birds have them and reptiles have them. Most reptiles have them. So understanding how a genome is organized at the sequence level, how big it is, and how it's compartmentalized, how it's organized into chromosomes, tells us a lot about the biology, the genomic biology of species. Here, for example, this is a beautiful graph showing the relationships, chromosomal relationships, between a human and a dog. So on the bottom part of this circle is the human genome, and it's organized on chromosomes. On the top is, on this part, is the dog genome, again organized on its respective chromosomes. So what this map is showing, it's a little complex, but what this map is showing is that this specific chromosome, in this example, that it breaks up and parts of it land in different chromosomes in the dog genome. What that tells us is that throughout evolution, chromosomes break apart and reorganize, sections of them flip around. And so it's not just that genomes are evolving at the sequence level, they're actually on the larger architectural level, they're breaking up and reorganizing and shifting all around. And that's important because it brings new genes in association with one another throughout the genome as evolution occurs. So one of the interesting parts of genome biology in terms of genome size is that genome size does not relate to organismal complexity. So a mud puppy, a Necturus, has a genome size of about 85 billion base pairs. The human genome is about 3.5 billion base pairs. Yet humans in many ways are more complex organisms than mud puppies, which is it's like a salamander. So for a long time, this was a big mystery. It was thought early on that the size of the genome should correspond to the organismal complexity because a big genome has a lot of genes in it. Well, what we now know is the situation is actually much more interesting. So this is a cell, and the little dots around it are a virus. And this unfortunate bacterium is being infected by a whole slew of viruses, and they're injecting their DNA into the cell. Now, what those viruses will do is they'll cause that bacterium they'll hijack the machinery and they'll cause it to make more viruses until oftentimes the cell will simply explode and all of the new viruses will go careening in the environment in this case, or in our bodies if we're sick. Well, we know now that during this process, we can have viral DNA get incorporated into genomes. And once it's there, it is just sitting along the string of DNA, like one of your genes. And these viral genes can copy themselves out and move around the genome. And by doing that, they can cause the genome to expand. So it was a pretty radical discovery with accumulating sequence data for humans in the Human Genome Project, that we realized that a large portion of our genome is composed of these kind of repetitive segments of DNA that duplicate and copy themselves around the genome. So these lines, that's a form of repetitive DNA. Signs, that's a form of repetitive DNA. And all of these different categories, transposons, the names don't matter so much, but these are different forms of repetitive viral DNA that copy themselves in our genome, and by doing so, they cause our genome size to inflate. What's interesting, if you look at the amount of our genome that's actually doing something, that's making gene products, it's this very small sliver. It's about 1.5 to 2% of our genome that's actually producing products, proteins that are building organisms, that are building our bodies. The rest of it is just kind of along for the ride, servicing its own evolutionary course. Now, despite the importance of these insights and what they tell us about life, we run into a great problem, and that is that about 99.9% of all animal species that have ever lived are now extinct. What this means is that we have, to understand genomics, to understand genome biology, we have a sample size of at best about 1.1%. And that's if we sampled all living animals. So we have a terrible problem in terms of sample size to understand genome biology, and that's caused by extinction in the fossil record. What we want to do, what I'm interested in as a scientist, is trying to go back to the fossil record and retrieve some bit of information about genome biology and trying to understand broader patterns of genome evolution and genome biology by bringing fossils to bear on hypotheses about genome evolution. So we're going to go back in time to the Mesozoic era and talk a little bit about some fossils from dinosaurs and about what they say about the genomes of birds. So first, let's have a frame of reference from which we can talk about these subjects. This is an evolutionary tree. It's called a phylogeny by scientists. Here is living birds. This is a red-tailed hawk. And its closest relatives were the carnivorous dinosaurs. This was the group from which birds arose. They're called the theropoda, the theropods. If you go down that evolutionary tree, then you hit the long-necked sauropods, and across the other group of dinosaurs, you see the Ornithischian dinosaurs. And these are things like duck-billed dinosaurs and Triceratops and Stegosaurus here. If you keep going down the evolutionary tree, then you will get to other reptiles, and if you go down far enough, then you get to other amniotes. And then if you go up, you get to things like humans and mammals. So during the past 10 years, there's been a real renaissance in understanding bird biology and how that's been informed by looking at the fossil record and dinosaurs. And how we can understand the origin of a lot of bird traits. So here, for example, is a bird that's sleeping. This is a common pose where they tuck their head behind underneath their arm, in their armpit. And this is a behavior that birds engage in that's thought to be associated with thermophysiology, with retaining body heat while they sleep. Well, we know now that that trait originated not in birds, but in dinosaurs. This was a discovery made in 2004, and it's an animal called mai long, which in Chinese means sleeping dragon. And you can see, this is a side view here on bottom, and you can see its neck is curved around. This is its head, and this is its arm, and it's in a sleeping posture with its head tucked behind its arm. So this shows, and this is not a bird, this is a small carnivorous dinosaur. And that demonstrates that the sleeping posture that we associate with birds is actually not a bird trait, that it was a dinosaur trait that the birds then just inherited. So birds also have feathers, like these beautiful feathers you see here. And feathers are a remarkable adaptation because birds use their feathers to do everything from flying, which you see here, to insulation, to insulate themselves in cold climates. And famously, birds also use their feathers to decorate themselves for the purpose of getting mates. So it was a really outstanding discovery, in about the last 10 years, where scientists discovered that feathers did not first arise in birds, but in fact they started finding them in small carnivorous dinosaurs, primarily from China. And this is a recent study of actually body size evolution in carnivorous dinosaurs. But this shows, I think nicely, here are birds over in this group. And this group, this phylogeny, this evolutionary tree, nicely shows that there was a high diversity in body size for these animals. Some were small, some were rather large, but almost all of them had feathers on them, whether they were birds or not, so whether they were flying or not. This means that the only real difference between early birds and some of these small carnivorous dinosaurs was simply because they were flying. But all the other traits that we think of, in early birds at least, were shared by these other carnivorous dinosaurs. Another trait that birds have that we're really interested in understanding as biologists is parental care. And penguins, of course, as are many other birds, they're famous for parental care. And we want to know how that trait arose. Well, paleontologists have found nests and nesting sites. They've CAT scanned these eggs and not only confirmed that they were that within the eggs were embryonic dinosaurs, but the scientists look at how the bone was developing, and it was clear that the animals were developing in such a way that once they hatched, they would have been helpless, which means that they had to have some form of parental care to survive. Here's, in fact, a remarkable fossil that demonstrates parental care even better. This is an oviraptor, and it was discovered actually sitting on its nest. These are its eggs underneath it, with its arms wrapped around its nest. So getting back to our main subject of genomics, we know many traits, organismal traits, things like feathers, flight, parental care. We know how they arose because we can look at fossils. But what about the genome structure of birds? How did that arise? Well, we can go back and look at fossils to understand something about genome size as well. Here's a graph that was first published by a researcher named Austin Hughes in 1999. And this shows the genome size. C value is another word for genome size. And this is measured actually in picograms in weight. So what these researchers actually do is they'll actually weigh a genome with very fine measurements, with very fine tools. So this is what Hughes did, is he broke down birds into 4 categories of how strongly they flew. Flightless birds, birds that fly weakly, moderately strong flyers, and then really strong flyers. And then looked at the size genome they have. Now, what he found is evident on this graph that the stronger flyers have the smaller genomes, and that flightless birds, in fact, have the biggest genomes. Now, out of amniotes, that is, 4-legged vertebrates who live on land, which is namely mammals, birds, and reptiles, birds have, out of that whole big group, birds have the smallest genomes. Not only do they have the smallest genomes, but there's not much variability in their genome. So they're small and they're uniform. They're uniformly small. So this is something that's unique in terms of avian biology, bird biology. And we want to know how and why birds have these small genomes. So what we can do is we can leverage a phenomena called the nucleotypic effect. And this is simply that the size of a nucleus or of a cell corresponds tightly to the amount of DNA in the genome. So to refresh your basic biology, a nucleus within a cell is a organ of the cell that contains all of the DNA for that organism. And so if the genome's bigger, the nucleus simply has to be bigger to fit the bigger genome in it. That consequently can cause a cell to become bigger. Here's an example of that relationship. You can see these large cells are aquatic salamanders, and they have very big genomes. Their genome sizes are about 83 picograms. If you compare those to human red blood cells, they're much bigger. These are human red blood cells for comparison. And human genome size is about 3.5 picograms. So there's a strong connection between the size of the genome and the size of the cell, and this corresponds nicely across a wide range of animals. So what we can do, what I did for some research that I have been engaged in, is I went and said, that's nice that relationship holds for red blood cells. Does it hold for other kinds of cells? So I went through and made thin sections of bone from a variety of different animals, and I looked at their bone tissue, and I looked at how big the bone cells were. So on top, this is a thin section of bone from a coyote, and the colors in it simply represent the direction of fibers within the bone. It's just a way for scientists to look at kind of the fiber architecture within a bone. They don't really mean anything for our purposes here. And on the bottom is a sample of bone from a rhea, a flightless bird. So we know we can measure the average size of bone cells, and we can also obtain the average genome size for these species. And then we can make we can look and see if they're correlated, if there's a relationship between bone cell size and genome size. And just like red blood cells, we find that indeed there is a strong relationship. Here you see a relationship between bone cell size on the bottom and genome size on this y-axis, where the bigger the bone cell size, the bigger the genome size. So what we can do is then we can use that relationship to try and predict how big the genome sizes were of extinct organisms like dinosaurs. So this is a thin section of a moa, another extinct bird species. And you can see all of these little pockets here are pockets where during life the living bone cells resided. So we can measure those and get an estimate for the size of bone cells. Here's another example of a thin section of bone from an extinct species. This is from a Tyrannosaurus rex. This is from a leg bone. And you can see again, these little pockets are where the bone cells sat during the life of the animal. And those structures are preserved within the bone, even though this is a fossilized bone that's over 65 million years old. So when we take those data, when we take the size of the bone cell size in the dinosaur, and then we take that statistical relationship, that correlation, we can use those two things to predict how big the genome size was in dinosaurs. We can then take that information and plot that on a tree, and we can color code it. And what you see here on this phylogeny, on this evolutionary tree, is a plot of genome size. So over here we have mammals. Here we have birds. Then here's a bunch of extinct dinosaurs. And over here we have reptiles and amphibian. And what you can see is that the living dinosaurs, birds, have purple, in this case for this map. They have purple-sized genomes, very small genomes. And our predictions show, our method shows, is that those small genomes existed in these carnivorous dinosaurs, suggesting that the small, unique, streamlined genomes of birds did not arise with birds, just like these other traits, like sleeping, like feathers, that there was a change in the bird genome, that it shrunk, and that happened in carnivorous dinosaurs, and that was a trait that birds then later inherited. Now that all the other dinosaurs are extinct, our only trace of that small genome, that small dinosaur genome, is found in birds. Now, as I mentioned earlier, what makes a genome big is the amount of these repetitive elements within the genome. So this is that same pie graph I showed earlier, these different kinds of repetitive elements within genomes. Now, it turns out that birds have very, very few of these genomic parasites in their genome. Consequently, that's what makes their genome small. So on this bar graph, this is a bar graph, and on the y-axis is the percentage of the genome composed of these repetitive elements. And you can see that these are mammals here, and for comparison, living birds have a very small percentage of their genome that's made up from these viral elements. If we move over and we look at what we estimate for these extinct dinosaurs based on our estimates of genome size, that they also had a genome that was very poor in terms of enrichment. It was very poor for for these repetitive elements, viral elements. And that this other main group of dinosaurs, the Ornithischian dinosaurs, were very reptile-like. It looks like they have an amount within their genomes very comparable to lizards and snakes and turtles.
We hope you've enjoyed this program and that you will join us for the next program in the series. For more resources, meanwhile, on fossils and ancient genomes, please visit our website and check the Resources tab. This program will be accessible on Belmont Media Center TV and other community television stations, and also on the Science for the Public website. Thank you.